It's my pleasure and honor to be here today. Thank you so much for everything you've been doing for us, and I'm very excited to give you an update on where we are on the data side. So looking at that question, by the way, 25%, if we'd asked this question only a few years ago, it would have been even much, much less than that. That tells you how much progress has been made fighting this disease, and how much progress is happening even every day. So with this, I would first ask our three panelists to join me here on stage. First will be Jonathan Keats from the Translational Genomic Institute. He's one of the bright lights out there and the brain behind all the molecular work being done behind Compass. Thank you, Jonathan. Next will be Dr. Ajay Chari from Mount Sinai. Ajay, you know, not only has a passion to treat patients, but he's also one of these, like many of our clinicians in our clinical network, the MMRC, also loves to put in Zens in the science and the data. And I must say, he also represents Mount Sinai, which is the single site that has put the most patients on the Compass study. Over 100 of the 1,200 plus patients on Compass, you know, come from Mount Sinai. So that tells you a lot. And finally will be Dr. Alessandra De Bacco. Alessandra is from Takeda. She's one of the wonderful translational lead at Takeda. She was very busy recently getting Ixazomib Ninaro approved last year for all of our patients. And also she represents a company, then Millennium, now Takeda, which is a long friend of the foundation, and also the first pharma partner who raised its hands to support our Compass study. So thank you, and with this, let's get going. Where's the clicker?
Oh.
It's over there.
Right there.
Thank you, Ajay. Let's start with this one, and this one here really represents, you know, why we're doing Compass. What's so special about Compass? These are two of our Compass patients, seemingly very similar, both Caucasian, female in their early 70s, intermediate risk staging, and both received the same exact treatment. They both have what would be called high-risk features, but the patient on the left progressed within a month, whereas the patient on the right is now still doing great over 2 years later. And this is really what's happening in cancer and in myeloma. No two patients are the same, and we need to figure out why, and we need to figure out what's the best possible treatment for a patient given where he is in his or her disease. So efforts like Compass, if you look below the dotted line, which is information that most doctors nowadays may not have, you will find, well, you know, all right, the two patients were deletion 17, and, you know, we've known for a while that, you know, supposedly deletion 17 is high risk, but we had some deletion 17 patient doing just great. So what's going on here? So through Compass, and Kathy presented that a little bit, and Jonathan can talk to this in a second, but we know that not all deletion 17 patients are the same. Going back also to this slide before, you see that one of these patients has more of these myeloma cells circulating in his blood compared to the other one. Much more, actually. And this is somewhat related to this microscopic disease, this even post-treatment, what we call this minimal residual disease, which we believe is a strong indicator of what's going on in a patient, and it could become a very powerful tool to develop new drugs. We'll discuss more about that going forward. But with this, let's start talking first a little bit about the molecular findings around Compass and the view on that from Jonathan. Jonathan?
Thank you. So my name is Jonathan Keating from the Translational Genomics Research Institute in Phoenix. It's nice to have an opportunity to talk to everyone for another year. I see some familiar faces and some new ones. One thing as we get into this, what I just wanted to point out quickly, considering who's in the room, if you're talking about where Compass is going, I'll just tell you my two quick views on it. One is there's very few places that have had the forethought to think a decade in advance. And this is a study where the benefits of the study are really going to show their weight. Year 7, year 11 is probably when the real benefit's going to occur. And it's also really important to understand one of the things that we're getting is not only the 1,000 patients, but when those patients do progress, they've already been put in place funding to look at 800 of those progression events. And I suspect we'll end up doing more or less depending on, if there's more that comes in the time, I suspect we'll look at them. But as those patients continue to come in, that's where the real differentiating factor for Compass is compared to all the other studies and other cancers. And that's why it is so important that the support that you can provide is what's going to keep this study from people saying, well, you got 1,000, what did you find? Shouldn't you have found something yet? It's, no, you got 1,000 and you're going to understand how the tumor evolves with time and how we treat patients better with time. And that's going to be what differentiates this study. So I do thank you for your support, but as they talk to you about things, it's important. What I wanted to show you here with this slide is just a very simple analogy that I usually tell people it's equivalent to getting your brakes checked. So this was really great when I presented it in San Francisco, because if you were to, somebody was to hop in a car with me and I said, well, the brakes are broken. And you might be smart enough to go, well, is it the brakes in the front and the brakes in the back, or both? And this is probably the simplest analogy that we should all focus on when we talk about some of these tumor suppressor genes. In this case, on deletion 17, the field has always been focused on a gene called P53, which we call the guardian of the genome, that it basically prevents cancer. So when we lose that gene, i.e. cutting the brakes, so we no longer have our brakes, that's a bad thing that allows the cancer to roll down the hill and take off. So a lot of myeloma patients today, we do a test in the clinic labs. Almost all centers would do this today, where they look to see if there's a deletion or is P53 missing. But generally what we've seen in multiple myeloma is that the one copy is missing. So we've cut the brakes to the back of the car. And if you were to hop in a car with me and say, well, I know the brakes are not working in the back, but they're still good in the front, you'd probably at least be nice enough and ride with me down to the brake shop to get my brakes fixed. But if I was to tell you that, you know what, I actually know the brakes don't work in the back and they don't work in the front, I don't think anyone's going to get in the car with me to go anywhere, even just to go get a cup of coffee. So in a myeloma tumor today, if the brakes are cut in the front or the brakes are cut in the back, we are telling those patients they're a high-risk patient. What we were able to come out with Compass is by doing the really detailed profiling that we're doing with that study, we're able to actually realize that, you know what, if you just cut the brakes in the front or the back, so just you still have brakes working on one side, those patients are doing as fine as the patients that we know have the brakes working in the front and the back. But those patients where we've knowingly cut the brakes to the front and the back, those are those patients who are doing so poorly. Now, where that's really important is in the clinic, that's about half of those patients that the clinic told were high-risk, poor prognosis patients. So if you're one of those patients, getting that information, half of them got the correct information, half of them got the incorrect information, and the physician also got correct or incorrect information, may or not been acting on that tumor correctly. So studies like this are now going to allow the clinics to change the way they do their testing so that they can include the proper testing so they can actually make that call. Do you have normal brakes? Are the brakes partly working, or are the brakes completely lost? Which will really help these patients and the physicians treating these patients. So that's one of those early events that we've been able to come out that I think at least hopefully everyone can understand.
So and the other one, Jonathan, you published one of these early papers a while ago actually, about this clonal tide in myeloma patient and how the disease in a patient is more like 4 different diseases, and how these different forms evolve with time, evolve with treatment, and that was very much put into the design of Compass. Can you speak a bit about that?
Yeah, so we had a previous study where we'd able to get every single patient that had ever been seen at Mayo Clinic, where we got a tumor sample from at one point in their disease and a second point in their disease. The entire Mayo system, that turned out to be 28. And we had one patient who we followed all the way through their entire disease course. And what we could see is how some tumors stayed the same with time, and some tumors really changed quite dramatically with time. Some even seemed to be changed by the therapy they got. So it really predicated us to want to know, are there certain patients who are going to be better set to get certain therapies versus other because the therapy wouldn't change their disease? Are there certain evolutionary things that maybe we can see early that if we saw that existing in a small percentage of cells, we would change the treatment to try and target those cells earlier before we let them take over the entire tumor mass? So with this study, we've had the luxury now, we've been able to screen 136 patients like that. We've had a couple of patients where we've had 3 or 4 time points. And it's allowed us now to start identifying common events that are coming and going with the specific treatments those patients were getting, which will allow us now to understand where are the things that are targeted for those treatments, so we could make maybe a better treatment that had less side effects. Who in here would like me to be able to tell you what we could make a drug that would work like dexamethasone but not have the side effects of dexamethasone? So as we get those kinds of information, that's going to be game-changing, because if we can really understand how the tumor gets around dexamethasone, we might be able to understand what it's really actually doing, and then make a drug that has much less side effects that gets us the same outcome. And studies like this are going to be so valuable for us to make those evolutionary steps in the future.
Okay, now Ajay, as a treating clinician, in your mind, what's the importance of Compass for our patients, and even for you in sometimes your daily practice? And Kathy, I know, showed some of the questions that are being answered right now by Compass, but let's get your take on this.
Yeah, I think as Kathy eloquently started with, we're kind of looking at cancer in a completely different way. And one of the best ways to explain that, I think, is comparing to non-Hodgkin's or lymphoma in general, which are related cancers. A lot of the drugs we use are actually overlapping. But for many decades now, we divide lymphoma into Hodgkin's, non-Hodgkin's. Then non-Hodgkin's is further divided into indolent, moderately aggressive, highly aggressive. So we have 4 basic categories. And the reason we've been able to do that is the pathologists are able to look under the microscope and tell us it's A, B, C, or D. And not just that, each of those patients gets treated completely differently. We don't treat the Burkitt's lymphoma the same way we treat some of the indolent lymphomas, which don't even get treated right away. So when you've been treating cancer from a pathologically slide-based approach, and again, you look at myeloma patients and it looks the same under the microscope, we've been stumped because the pathologists haven't been able to help us. And finally, we have this era of genomic medicine where we're going to be doing better than what we've done, and I think we're not going to be treating everybody the same. But in order to do that, and I think what makes Compass so important is the genomics is super important, but genomic data will be useless without the clinical data, because you need all of that component. We talk about risk, but the genomic risk is one component. There's the host factors. So you can have an older patient who may not have any high genomic risk, but they're not going to live as long because of the complexity of their organ systems and other issues. There's the disease factors in terms of the burden, radiologic, how much disease is there, any spinal disease, and then of course the outcomes with treatment. In some ways, somebody who might be standard risk, but we don't yet know why they're high risk and they have very short remissions, is also high risk. So you need that kinetics of treatment response, the host factors, the disease burden integrated with the genomic data, and then you can really start to get at who's high risk, low risk, and treat people accordingly. But I think in order to do that, I don't know if you have those other slides.
Absolutely. Those were shown. We'll come back to this.
So this is our current armamentarium. The columns on the left was drugs that we've had for many years, but those next 4 columns, IMiDs, proteasome inhibitors, HDAC inhibitors, monoclonal antibodies, all of those were only approved in the last 10 to 15 years. And you look at this and say, wow, we have a lot of drugs. And some patients now, this may be enough. There are some patients where there have been publications that they have no relapse in 10 years. So can we use the current arsenal to figure out who can be really treated adequately and when do we stop? That's, I think, really important that patients often ask is, when can we stop therapy? And the problem is we never have studies that do those kinds of questions, because if you don't know the different types of myeloma, everybody gets treated the same. And all we ask is 2 drugs versus 3 drugs. But to ask stopping early versus continuing, that's going to require not only identifying the patients at different types of myeloma, but then different stopping endpoints. So then how do we move past this? Because clearly this is not enough for a large number of patients. And then on the next slide, I think we have...
I incorporated some of your own questions in that slide.
Okay. So I think the question is, so for the current COMPASS questions that you could ask, you know, right now we talked about it's hard to know these different kinds of disease, but right now with those 1,000 patients, if we asked Dr. Keats and his group to say, I'm not going to tell you different clinical profiles, tell me if these patients now segregate. So we could ask about, we have patients, our youngest patient at Mount Sinai was diagnosed with myeloma at age 18. Is that the same myeloma as the patient who has it at 90? And can you genomically differentiate patients who get myeloma below the age of 40 versus those who get it in their middle years versus those who get it in older years? Similarly, you could ask that question about race. You could ask that question about different clinical presentations, who gets renal failure, who presents with bone disease, and who doesn't. And we've also talked in COMPASS that the way that COMPASS worked, I mean, again, out of the graciousness of patients, many patients were willing to undergo repeat biopsy, even though they'd had an initial diagnosis. But some patients had their first biopsy banked at COMPASS, and it turned out that they were felt to have smoldering myeloma and didn't get treatment. That's a huge gold mine of patients that we can now compare to. Why is it that some patients had symptomatic disease and others didn't? And can we better understand? We've done these in small academic center efforts with maybe 20 patients, but this is going to be unprecedented when you have 1,000 patients with symptomatic myeloma who've gotten treated, and we now know there's about 200-something patients who volunteered to participate in COMPASS, but they never got treated. And that's data that we're sitting on, and we need to go back and compare what makes these patients different. And that's going to get at the prevention question. How do you, if you understand what caused some patients to convert to symptomatic disease, can you then intervene early? And so those are the kind of questions we can do just from an initial sample time point. But then to really get at, I think, the future, which was how do we move past those 10 drugs, is what happens to these patients who progress? Why do some patients progress sooner and others later? And what's happening genomically? So because if we can understand why the current drugs aren't enough and why the cells become resistant, that'll help to generate the next wave of medication. So I think there's really two important things that COMPASS gives us. The initial profile, but equally important is the clinical annotation as patients evolve over time. And I think those are going to be huge, important considerations in getting past our current hurdles. So I'll just stop there.
Yeah, it's so well said, Ajay. Thanks so much. So Alessandra, you know, as one of these translational lead at Takeda, you've been using a lot of, you know, clinical genomic data to move drug development forward, including with ixazomib and Ninlaro, and you've also been looking quite a bit at COMPASS data. What is this kind of information does for a company like yours?
Yes. So I would like to start just to say that among all the floods of genomic data that are available nowadays, the COMPASS dataset really stands out in the way it came to be, really, because it was thought-driven and it continued to be led by patient, which is very different from all the genomic data that are produced by institutional, including pharma. We have a very large data set as well from our own clinical trials, so it has the uniqueness. But what I found particularly valuable in COMPASS is not only this extremely deep understanding of the genomic, but the association of the clinical data set for what is the standard of care today. Because the data that come from a clinical trial are data on the population enrolled in the clinical trial. What COMPASS produce are the data and the efficacy of therapy in the real world. And so you can really understand the depth of response. You can really understand the duration of treatment of the therapy that are out there. And you would be surprised, sometimes are not the same length that you normally see when you do a testing, but just that is an intrinsic difference in the way you test a drug in a trial versus when you use it every day in a clinic. So those information are absolutely priceless. And also most of our trial and also of other pharma, you do analysis of the patient before actually get treated. So you have the genomic understanding of the tumor, and then you connect with the clinical data after treatment with your drug. But we don't have, we ask sometimes, and that's totally an optional sample, to get a sample relapse in order to understand what happened to the tumor that bypass the activity of this drug. And that are very critical information, because if there is a mutation, if there is a pathway that get activated, then we needed to know what to do for this patient. And unfortunately, those are very difficult samples to get, so we never really have enough. But with the design of the continuation study, that has a tremendous value. And we as a Takeda, we really kind of supported that, and we are very happy to partner for this continuation of the study. But as you said, what we do is with our team, we try to understand what are really the patients specific for our therapy, that benefit the most for our therapy. We try to confirm our observation in a completely different data set, because that is very important. And luckily, the Compass has already a lot of the data from our previous compounds, so they have a lot of similarity, and so we can extrapolate about some of our observation has been just happened in this trial versus something that really merits an additional look. And also has, as it was described before, the opportunity to look of the immunosystem repertoire. That's something that is super valuable, because even patients that don't achieve complete response can maintain a stable disease for a long time. That means that it's not only the tumor, it's the entire system that in some way kind of coordinated. And we need really to understand that part. And the other component is the quality of life. A lot of time, nowadays we have more and more drug for multiple myeloma, but we need really to understand what's the burden on the patient. And if a therapy is beneficial to you, but you cannot have it for a long time, that at the end is a therapy that does not help you. So it's very, very important to understand the features of the therapy, the characteristic, and not only the adverse event, but also how the patient live the life. Because you want to make a disease first chronic and then cured. In order to contain the problem. And so we have been using enormous time even to see how a patient that originally was not high risk became high risk. And we always, most of the time in our study, you use the participation are patients that have 1 or 2 relapse, 3 or more relapse. But the reality is that each of these relapses are very heterogeneous because they come from a very different therapy. So if you think the disease has a set of balls of different color, not all the color are represented in the same amount. So some tumors have more red, some other a little bit of blue. And when you try the therapies, if therapy eliminates the red, you end up having predominantly blue at the end. So you need to know how the disease evolves under the pressure of the therapy. Because at that point, you know what can you do afterwards with the patient. And so that's extremely focused. We do in our own study. We do a lot with the Compass, and actually there is a group in our company that looks at modeling. So what they wanted to do and see, looking at the kinetic of the M protein over time, for a certain period of time, how can you predict the behavior of the disease? In this way, just an idea to provide, for example, a tool for physicians that might not be in academic center, that might not treat very often myeloma. They might have like just a handful of myeloma patients, and as soon as there is an adverse event or they don't see a certain type of response, they don't know what to do, and just withdraw the patient from the therapy. Just give them a tool to say, that's fine, that's what happened, but that's what is going to happen later. Right? So we are trying to use some of the data, longitudinal data, to really model and help provide a tool to physicians to try to understand what to expect on a disease as it progresses, as it goes through the different phases of treatment, and so that it can help the physician better. And then last, I would like to conclude of just to say that with MMRF have done a tremendous work, create a non-competitive space where all the pharma can come and work together. And nowadays more and more pharma gets into this industry-wide partnership simply because the burden, the economical burden of the basic research is so high. And unfortunately, there are not as many products that come out at the end. It's very, very difficult to develop, identify new target. So there is this opportunity to come together and really work together for innovation. And everybody is really aligned on that goal and really moving forward and do something for patients. So absolutely appreciate that.
Thanks so much, Alessandra. So many interesting points raised there. So I'd like to go back to a couple of them. You mentioned quality of life, and in Compass we have well-built quality of life. And it goes back to what Kathy mentioned earlier. In the old days, patients used to be simply human subjects. Yeah. Now, more and more, we're making them active participants in the research. And through the very thorough quality of life data that we have in Compass, for example, those can even be important to predict adverse events. For example, we were looking at patients who, on a scale of 10, were reporting 10 out of 10 for severe constipation, for example, which is a well-known side effect of IMiDs. And what we were seeing is that there were many more of these patients reporting that side effect than what the clinician had been reported for the same patients in the Compass study, which is not so surprising. We know that patients sometimes tend to over-report, but then clinicians tend to under-report. But then when we went back to these clinicians and asked them, so how about this patient? How about that patient? How about that patient? It turned out that every single time the clinician said, yeah, maybe we should have reported that. We should probably have reported that one as an adverse event. So even the data coming from patients can be an harbinger of adverse event to come. So in some way, the patient themselves become really participant in the effort. The other part that you mentioned, Alessandra, about these relapsing patients, they're coming from so different many types of treatments and approaches, and they're coming from so many different angles. So likely 1,000 patients won't be enough. It's obvious, but I'd like maybe to point that out to the entire group here. So, we at the MMRF feel that we need each and every one of us to cure each and every one of us. So why, do we need to aim to have the same kind of gold standard treatment given to Compass patient to every single patient? And why should we expand that at least countrywide, if not worldwide? Maybe Ajay, you want to start on that one?
Yeah, I think you bring up a couple of points. In physicians, we typically look to published clinical trials, prospective clinical trials to guide our management, which are very important because you standardize the intervention and you're selecting a particular patient group. But some of the limitations to that are you miss out on real-world issues. The frail, the elderly, the people on dialysis, the people with multiple other organ problems. Those patients are never entered in clinical trials involving a new agent. So you miss out on that part of the data. And also, I think their access to drug differs globally. So that's another important thing. And you also alluded to quality of life issues, which, typically in clinical trials, the way they're reported is you look at mild, medium, and severe, and we usually focus on the severe toxicities. But many patients would argue that low-level severity, but chronic, in some ways is much worse than maybe a severe short-term side effect. So I think there's a lot of stuff that clinical trials leave, and we're left with gaps in our knowledge. And I think but it's not either or, it's both. The knowledge we gain from Compass can then go back to query prospective clinical trials where you really do have very controlled, 2 drugs versus 3 drugs. And if your hypothesis is that this target mediates the mechanism of resistance and you can validate that in the other cohorts, there's going to be a back and forth. But the problem with limited clinical trials, you're only getting a few drugs to look at and a very small selected group of patients. So I think that's the beauty of Compass.
Yeah. Any other thoughts from the panel? Maybe Jonathan?
Is the one thing, when you're talking about how many patients do you need, that we've learned, we did the Multiple Myeloma Genomics Initiative with 250 patients. Now we're well, we had 925 patients actually studied in Compass and soon to be almost the full 1,000. As you do more, you start to, the biggest thing you appreciate is the people who do statistics have been right all the years, that 200 is not a very big way of actually sampling what the disease looks like for the average patient out there. 1,000 probably isn't. There's always limitations, but obviously things that would attempt to understand a larger and larger patient population gives us a better and better understanding. The nice thing is in the field with what the MMRF has sponsored and other groups have sponsored, we are talking in this field now about having really good characterization data on thousands, probably 2,000 to 3,000 patients. So we're getting to the point where even the really hardcore statisticians would tell us we're getting close, but some people will tell you that it needs to be way higher than that.
Yeah. Thank you. So let's focus ourselves on a couple of questions we're getting, and please, everyone, please don't be shy, send out questions. And one of them actually is relevant to something else we wanted to show. I'll punt that one first to you, Jonathan, and then to Ajay. Does Foundation 1 evaluation for mutation count as tumor sequencing? And I also want to point to something somewhat similar we're doing to our relapsing patient within Compass. But Jonathan?
No, I think things like Foundation 1 or Foundation 1 heme have a lot of advantages. In the case of Compass, all Compass patients, when they progress, are getting a test done at the clinical lab at TGen that is very similar to the Foundation heme test. This is one of the examples. It's actually not my favorite one, because the favorite one I like to kind of present to people was almost everyone's heard of the breast cancer gene. So there's a series of drugs that have been designed specifically for people that have the breast cancer gene, which in multiple myeloma, it's not breast cancer, we would never look for. But actually the third patient that we profiled on relapse was the molecular basis for why they developed a drug to treat patients that had the breast cancer gene. So we sent out a report to one of the treating physicians groups saying this patient would be the prime candidate for that type of event. Now, similar here with RAS or ALK, we can also identify things, but I think for patients, these things do have value. Maybe at diagnosis, I'm not really sure how much justification there is to use them, but definitely when a patient progresses, I think the more we can provide options to the treating physician, the better the patient outcomes should hopefully be.
So Ajay, some thought on that front, especially when you're getting these patients completely running out of options. This is going to be a discussion we're going to have at a later panel this day, but since we have, you've put so many Compass patients on this trial and are starting to get these kinds of reports. What's kind of your reaction to those, and what are thoughts going through your mind when you start seeing these reports?
I think it's another important tool that we have in our patient management. I found that it's been perhaps the most useful, and we've been kind of alluding to these clonal tidings or, the balls with different colors, but we've seen patients where you treat them on a novel agent combination, they have an excellent response, but then there's maybe this one clone that's growing, and that's becoming painful, and you biopsy that clone, and you find a mutation like this, and you can target that mutation. So it's been very helpful in that sense, but I think one of the limitations of that is right now, Foundation's been only doing, they're not doing any CD138 selection, meaning we're just getting a sample that tells us the mutational analysis of the specimen sent. But if we're really going to get past these kinds of hurdles, we need to look at, I think, all three components of the genomic sequencing, which is not just the genomic, but the entire. So you have to look at the myeloma cells, the immune environment, and then the patient's own baseline genetic predispositions that may be not even present in the cancer cells. So I think we need to integrate all of those three components. And one of the limitations of these testing is that you really only get something, and if it's a biopsy with a lot of myeloma, you can be pretty confident that that's the mutation in that clone. But if it's a blend of different cells, you may miss it if you're not doing the enrichment for the myeloma. So I think we still have some technical hurdles, but also biological hurdles to get past with this.
Yeah, absolutely. Maybe somewhat related, here's another question. Here's a patient who's been treated at a site and is wondering, I have no idea whether or not my tissue samples was contributed to a sequencing study or not. And that's a big question, by the way, there's a lot of data. Again, this is why it's important that patients become part of the solution, become part of the process. Many of you patients probably, did have some form of molecular profiling done, but you're not even aware of that, and it's kind of sad in some way. If you were part of a study that Compass, you will know, absolutely. But this is where, again, building going forward a more direct-to-patient approach will enable each and everyone to know exactly what has been done from a testing perspective on them, and help to contribute that data to this greater myeloma community so we can learn even more as we're learning every day out of Compass.
Can
go
I
ahead,
add
Alessandra.
actually something to your point? And I think I see the mutation information of the tumor as a kind of a library, right? That will add information to the larger Compass, but also is a library that whatever there is a new therapy targeting a mutation of genes that was not done before, then you can just go back and just say, oh yeah, I had that at that point. It depends on how long it was before. So it's just having extra cards in your sleeve. You might not use them all now. But having the possibility of accessing them retrospectively. Because unfortunately, we don't know what to do with all the mutation that we found, but maybe more they're going to come, in the future. And I think that is important to leverage your information that already exists. And I think that could be a good use for that. And definitely, if a sample was taken, you should know if it's been sequenced, because it should be in your informed consent. Well, that's what's being used.
We should add that if you were not a patient who was able to participate in Compass initially, we now have through the MMRC, through the University of Michigan, an opportunity to sequence your tumor at a relapse time point as well. So
absolutely. There's a number of questions around precursor conditions, smoldering. So those we'll keep for a session this afternoon. Especially, we'll make sure we have these questions we put to Irene when we have this panel. So any maybe finishing thought on why studies like Compass are so important, and how it's so important to be more precise than we've ever been while treating our myeloma patients. Maybe start with Jonathan.
Well, I guess one, to give you the impression for how important this study is, it's always easy I can give you the example of what my peers say when I go out and give a talk and you say what the Compass study is. And most people kind of first kind of give you you're joking. And then they say, you're doing what exactly again? And you explain the second time. And then their next response is, who's paying for this? And that response is the shock, I don't believe you, because no one can do that, because that's just way above anything you can do. The second one is okay, now I'm actually starting to believe you, and I want access to who's paying for this. But it shows you the kind of forethought that the organization has had, that you can't plan small if you want to have big effects. And I'm a big believer in that. Don't dream small, because you'll get small rewards. Dream big and you'll get big rewards. And that's definitely what we do here.
Well said.
All right, let's say, Ajay, if you can one-up this one.
I'm not even going to try. I think the main thing we do in medicine, though, is at the end of the day, if you have to say what we do as physicians is optimizing risk and benefit. And that's what precision medicine is all about, right? We can take the same drug and give it at the same dose and schedule and frequency for every patient, but that's not going to be the right way to do that. And so in order to optimize risk and benefit, you need to know the patient's risk, you need to know the therapeutic intervention's risk, what the benefit is required, and balance those two. And then the other thing that we have really very little clue to is sequencing of drugs. How do you pick the right sequence of drugs? And we need these kinds of studies to understand what's happening to patients, because we keep getting moved, take 1 drug, 2 drug, 3 drug, probably we're getting close to 4 drug because of the monoclonal antibodies. But then what happens after that? And what's the next drug after you've used up those 4 drugs? So I think that's what I think Compass can help us do, is really to do precision medicine, you need to optimize risk and benefit in a scientifically based manner. And that's what Compass will help us do.
Absolutely. Alessandra?
So I have already mentioned many of the values that the Compass study is provided to the patient, to the physician, to the community, and also to pharma as well. But what I particularly like the Compass is what MMRF has achieved, which is something that I think is important for any disease, is realizing that each individual of us will not be strong enough to develop medicine to treat a patient. Each of us comes with a different strength. And so it's only working together as a patient, as a caregiver, as a pharma that develops and try different drugs and programming. It's only that's the way, and that should be a kind of methods that is applied to any other cancer. And that is, I think, the most powerful, to be able to show that you can have all this part, although each of them has like a different interest, but they come together because they are unified in the ultimate goal. And so that is possible, and you guys are doing. So I think it would be a nice model to replicate in other type of disease, and I'm sure that will be as successful.
Yeah, I think this is the way drug discovery and cancer management will be in the next 3 to 5 years. I think we will stop here. I will give Anne a chance to provide some final thoughts on this first panel, but I would like to thank all 3 of you. Again, thanks so much, and thanks for your questions out there.
Yeah, I do want to echo Daniel's thanks to the panelists. I think all of us at the MMRF and hopefully everyone else in the room just feels so grateful to have such, compassionate, passionate, committed partners from pharma, academia, research with us in this effort, because as Alessandra said, it takes all of us. We all have unique strengths and we need to come together to realize it. A couple points I really wanted to emphasize that I thought were incredibly important. I just wanted to make sure that they weren't passing comments in the panel that got lost. The first one is what Alessandra mentioned about having this real world data. And one thing we didn't mention to you today, and some of you may be aware, is that in Compass, in those 1,000 patients, we have patients as young as, I think, 19 all the way to 91. And we have almost 20% African Americans. We really have a representation of what multiple myeloma looks like across the United States and some of the European and countries in Canada that are represented as well. Clinical trials aren't that represented. They typically have younger patients, a predominance of white patients, patients treated at academic centers. You need to be healthy for a lot of these trials. So you're not getting the full picture of what real world myeloma patients look like and how they respond to treatment. So it was very important for us to do that and to make sure that we were getting an accurate snapshot. And it also gets to the point then of, as Jonathan said, well, who's doing this study? Who's paying for it? And you can see why one company really wouldn't be incentivized to do this, especially, unfortunately, for a relatively uncommon disease like myeloma. Certainly not the government. We didn't wait around for the government, 12 years ago for the first genomics initiative. It took an organization like this with the support of the companies who provided some of the funding and all of you to take this on. It really had to be us, and I think the panel really reinforced that. A couple of other things. We focused a lot on high risk, and of course we're learning about high risk. One of the first learnings coming out of Compass is around high risk patients because they relapse faster. But Ajay, made the point too about patients who may not need as much treatment, patients who may be able to stop treatment earlier, even though in general patients on triplets did better than patients on doublets. Well, who are those patients on the doublets that did just fine, who don't need that third drug? So this is the type of information that we'll be able to learn from Compass, not just who needs more, high risk, maybe who needs less. And then finally, I think what is important too, and Ajay kind of made this point, when we put together this program, and in the next panel you hear more about the gateways where the data is made publicly available, it was always our vision, maybe a little simplistic, but one day in the future, a doctor could sit down with his or her patient and say, okay, you're diagnosed with myeloma, you have these characteristics. Let's look at the COMPASS database, find patients who look just like you, see what kind of treatments they were on, see what they did, and make rational treatment decisions based on that kind of data. So it'll take a while, as Jonathan said in the beginning, for the data to mature, but that's what we're hoping to learn. So that patients, perhaps some of you in this room, and certainly patients for the future, will have a very clear path in terms of the sequence, the combination of treatments that they should be on. Thank you.